Biology 2121, Presentation 12, Sensory Systems. You'll remember that the peripheral nervous system consists of motor pathways and sensory pathways. And the motor pathways, which we had also referred to as efferent pathways, are nerves that exit the central nervous system and head towards effectors. And those effectors might be muscles, and those effectors might be glands. And we did a pretty good job at identifying how those efferent neurons innervated muscles. And we did that in the context of the material on muscles, and we also covered it a little bit in our material on how neurons actually work. But we didn't really spend a lot of time on the sensory portion of the somatic nervous system. These are our receptors, and these are afferent neurons bundled into afferent nerves that carry the sensory information into the central nervous system, where it will be integrated and it will be routed outwards to the appropriate effectors through the appropriate efferent neurons and nerves. So we're going to talk about sensory portion of the somatic nervous system. And we're going to begin with this slide on sensory integration. You see, the thing is when we touch something or we perceive temperature or we have baroreceptors that identify changes in pressure, all that information is carried into the brain. It's detected by the receptors and it's carried into the brain in terms of a series of action potentials. And you really can't distinguish between one action potential and another. We learned way back in our first presentation on neuron function that action potentials all operate at the same basic intensity. And so you can't essentially change the nature of one individual action potential. So I'm thinking about, for example, perception of touch. That perception of touch will send a series of action potentials traveling down my afferent neuron towards my central nervous system, and I can't differentiate between one and the other. What's going to happen, though, is every single one of those action potentials will show up on the doorstep of my brain, and at that point, my association or integration neurons in my brain will receive that information and that point make a critical decision in terms of what it actually means. So it says here, sensations are detected and carried to the brain. They're detected by sensory receptors, and they all send the same type of electrical signal, action potentials. But at some point, that receives our brain, and that's where we execute what we call perception. Is it rough? Is it smooth? Is the pressure too high? Is the pressure too low? Is it bright? Is it dark? Is it loud? Is it soft? All these different variations that we understand constitute details of our perception of sense are constructed by our brain. And so we depend on the fact that our brain is doing an adequate and accurate job in interpreting that. So perceptions are constructed by the brain. Perceptions are degrees of intensity of a particular stimulus. And every single one of us perceive these a little bit differently. Its integration is a consequence of sensory input, and it generally involves multiple brain region communication. So for example, when you smell something, that sensory information is going to head into your olfactory sensors in your brain, where you'll interpret the smell, but a lot of times that's also going to connect to emotional regions of your brain, because a lot of times we associate smells with, kind of reactions we had with things in the past, and might actually connect to memory centers in the brain as well. Gee, I smell that. That reminds me of the smell of the lunchroom when I was in first grade, things like that. And so sensations and perceptions, make sure you know the basic difference. Now, a little bit later, we'll specify that one important aspect of determining degrees of intensity, and that would be perception, has to do with the number of neurons being stimulated. The more neurons, the greater the intensity of the sensation. Could have to do with the frequency of the action potentials being carried. the more frequent the action potentials, the more intense the perception. And then sometimes the individual pathways will have an effect as well. And then, once again, the final arbiter or the final decision maker on all this is going to be those association neurons in your brain that are connecting with these multiple brain regions. And that's ultimately going to determine your perception of that sight, that smell, that sound, those details of monitoring things going inside of your body, like body position and balance and muscle tone and that sort of thing. That's all sorted out by your brain. And at that point, your brain makes decisions on how to deal with that. You had a bit of an introduction to sensory receptors when you covered content on skin anatomy. And you'll remember in the context of skin anatomy, the dermis is a very rich, structure-rich place in the skin where I got all kinds of blood vessels and glands and nerves and sensory receptors. Now, sensory receptors, as a rule, fall into three general categories. They're illustrated here, and you'll need to know those three general categories, and you will definitely need to know the representative examples. This particular illustration is taken from your textbook. Free nerve endings, you'll see that illustrated at the top illustration, are representative of pain receptors, also known as nociceptors, or I prefer the pronunciation nociceptors. Then there are encapsulated nerve endings like Meissner's corpuscles, which you should be familiar with in context of skin anatomy. And then finally, specialized receptor cells like photoreceptors that are going to be involved in vision or sight. For example, rods and cones found in the retina of the eye. So we can classify sensors into three basic types in terms of more or less what they look like. But we can also classify sensory receptors in terms of their proximity to the stimulus. Are they near? Are they far? Where are they located? And then there will be a whole bunch of other distinguishing terms that we're going to be using to identify sensory receptors. So the next two slides added to the previous slide are critical in terms of being able to identify and characterize the specific types of sensory receptors you might encounter when looking at the human nervous system. These three slides, number 3, number 4, and number 5 of this presentation are going to be critical for you to know. Okay, so our next way of categorizing sensory receptors is proximity to stimulus. Exteroceptors are receptors that are located near the stimulus, primarily right where that stimulus is being generated. So for example, if you touch something, the exteroreceptor is right there where the touch is occurring. If you experience pressure, the receptor is right there where the pressure is being placed. If you experience pain, the receptor is right there where the pain is being generated, and temperature right there where the temperature is being perceived. So exteroreceptors are proximal or right near the stimulus. In contrast, we have interoceptors, which are receptors that are going to be located a little bit distally or distant from the stimulus. For example, interoceptors are going to be monitoring things like chemical messengers traveling through your bloodstream, maybe ions, maybe sugar content, maybe body temperature, maybe blood pressure. So technically these things that we're monitoring aren't necessarily right there. They're more distal and certainly more widespread through the body. So exteroceptors are right there. Interoceptors are monitoring things that are a little bit more distant or distal. And then finally, we got proprioceptors. And these are receptors that are going to be located near moving body parts. We mentioned once upon a time that proprioceptors are really, really important in monitoring body position and balance. And proprioceptors are going to be monitoring position and stretch of joints, tendons, and muscles, leading to our brains being able to interpret our particular position, our posture, and our balance at any given moment. Here's our third slide on the classification of sensory receptors, in this case, functional classification. What do they do? We got nociceptors or nociceptors, which detect pain, thermoreceptors that monitor temperature, photoreceptors that detect light. Mechanoreceptors are probably the most numerous receptors found in the body and contribute probably to some of the most important things that we need to monitor, which would include blood pressure, would include stretch and position of our muscles and our bones, auditory receptors, which is how we hear, and balance receptors in terms of the vestibular system, in terms of how we maintain our balance. Mechanoreceptors detect physical stimuli, such as pressure, vibration, or position. Then there are chemoreceptors, which are going to be those receptors that are molecule specific. They react to the presence of certain molecules, resulting in an interpretation of taste or smell. And then finally, we have osmoreceptors that measure solute concentration of bodily fluids, primarily fluid in the bloodstream. And that is sometimes referred to as osmolality. That's OSMOALITY. Osmolality is a very commonly used term to indicate the concentration of solutes, dissolved substances in the bloodstream. And the higher the osmolality, the greater the movement of water from tissues into the bloodstream. And you could also measure osmolality in tissues as well, which could similarly drive the movement of water from the bloodstream into those tissues. So osmoreceptors are going to be critical in monitoring salt-water balance in the body. Let's talk about the sense of taste, officially known as gustation. And taste is associated with the presence of physical structures located primarily on the tongue called taste buds. Taste buds are structures that have numerous taste cells or taste receptors located on each taste bud, and those receptors are chemoreceptors. See, we encounter various chemical compounds in the foods we eat, and those chemical compounds trigger depolarizations in those receptors. And those depolarizations are carried to our brain, which interprets those as taste. Is it sweet? Is it sour? Is it salty? Is it bitter? That sort of thing. So on our slide here, we're going to understand that our taste receptors found on our taste buds can detect 5 different chemical types of taste. And those tastes qualify as bitter, sour, salty, sweet, or something called umami, which is associated with the presence of protein in the food that we're eating. And it's also sometimes described as a savory taste. Water is regarded as a 6th type of taste. So technically at this point, I guess we say there's 6 different chemical types of taste: bitter, sour, salt, sweet, umami, and water. As stated before on the previous slide, you got anywhere from 50 to 100 taste receptors on a single taste bud. These taste receptors come in a number of different types depending upon what sort of thing they're receptive to. For example, take a look at a slide here. For salty and sour tastes, we're going to depend on taste receptors that depend on chemically gated ion channels. Something that's salty is going to be essentially ionic. Something that's sour is going to be characterized by the presence of hydrogen ions, i.e. acid. So those ions, whether they're hydrogen ions or sodium ions or chlorine ions, they're going to open chemically gated ion channels. That's going to establish a depolarization in the receptor cell. Once that depolarization takes place, the receptor cell can then release neurotransmitters, NTs, which will then depolarize any sensory neuron and will carry that information into the gustatory cortex of the cerebral cortex in the brain. Hence, we will interpret that as being a salty or sour taste. Turning our attention to sweet, bitter, and umami, in these cases, the receptor is going to pick up on the sweet, the bitter, or the umami. That binding of the chemical compound that creates that particular taste is going to activate the release of a second messenger within the receptor cell. That second messenger compound might be something like cyclic AMP, but regardless, what it's going to do, it's going to stimulate, once again, release of a neurotransmitter, at which point will depolarize a sensory neuron that's going to carry that information into the gustatory cortex of the cerebral cortex and will interpret that taste as being sweet, bitter, or umami. And then finally, water, as our sixth taste, is where water molecules enter through a specific type of channel protein found in our cells called aquaporins. Aquaporins are critical to the process of osmosis. So water enters through aquaporins. The name aqua, water, porin, hole gives you the idea, yes, this is a channel protein that's going to permit the movement of water. Well, as soon as that water moves into the cell is going to swell. It's going to increase in volume. Now, my third bullet point here says triggers volume-gated ion channels. Probably a better way to think about it is these are like mechanically gated ion channels. We talked about mechanically gated ion channels way back when we were talking about neurons. And the fact of the matter is that when volume changes in a cell, that's going to elicit some kind of mechanical change in the membrane, which could therefore then trigger those ion channels. But as usual, regardless, the ion channels open. We get a depolarization event. That depolarization event releases neurotransmitters, in which case we will effect a depolarization in a sensory neuron, and that information is going to be conveyed to the cerebral cortex and therefore be interpreted as the taste of water. All right, a lot of people get confused. When you take a look at your tongue in the mirror, you see all these little bumps all over it. And a lot of people think those bumps are taste buds. They're not. Those bumps are called papillae. Those papillae contain those aggregations of receptor cells that we call taste buds, and those taste buds are embedded within the papillae. They're attached to their respective sensory neurons, and those sensory neurons enter the tongue and then find their way ultimately to the central nervous system and to the gustatory cortex portion of the cerebral cortex. Now, taking a look at these papillae, the papillae are classified into a number of different types based on shape, in part position, and at least in one case, there is a type of papilla that has no taste buds on it at all. And that particular papilla is more concerned with detection of the fact that the food is in the mouth and the consistency of the food. And obviously that combination of taste and consistency all equates to this overall sensation concerning what you're eating, what it feels like, and what it tastes like. And those combinations will essentially determine whether you want more or you want less, because your brain interprets that as not just feeling good because you're eating, which is called appetite, but in terms of determining whether or not you get the nutrients that you need, that you require. Are you getting the calories? Are you getting the fat molecules? Are you getting the protein molecules? That's what constitutes hunger. Hunger is a physiological drive to eat. Appetite is a psychological aspects of that. So let's take a look at the classification of papillae that's demonstrated on this slide. I see an illustration of a tongue, and I see a number of arrows indicating some close-up views of papillae that you need to be aware of. There are fungiform papillae, there are foliate papillae, and there are circumvallate papillae that are all going to be concerned with taste. You'll see they differ in terms of their appearance. And the one I would like to focus on are the circumvallate papillae, because the circumvallate papillae are the largest of the papillae, and they're generally found further back on the surface of the tongue. You take a look in the mirror and you stick your tongue way out, and you'll see these weird looking bumps on the back of your tongue. And some people get concerned and say, well, golly, those must be some kind of tumor or something, some kind of growth. No, those are circumvallate papillae, and they are found at the posterior region of the tongue. The circumvallate papillae have taste buds. The foliate papillae have taste buds. The fungiform papillae have taste buds. These papillae are critical to our sense of taste, but there's one more papilla I need to mention, and that's the filiform papilla. The filiform papillae are actually the most numerous. They cover as much as 2/3 of the tongue's surface. And rather than being concerned with taste, the filiform papillae are much more concerned with the texture and the feel of the food in the mouth. And obviously that's going to contribute a heck of a lot to your ability to chew, because chewing, or mastication as it's officially known, is in a large part, yes, due to your teeth, but it's also due to the coordinated activity of your tongue in making sure it's pushing that food into the pathway of your teeth. And so there's a whole lot of stuff going on in your mouth. It's not just taste, it's not just chewing, it's not just coordinated activity with your tongue, it's also sensing all this stuff, tasting all this stuff, and making sure that you know where it is in your mouth at any given time, and are you getting the food molecules that you need. That is the combination of things that's so critical as far as all these sensors located in your mouth. So taste buds actually decline with age. I mean, as you get older, you start losing receptor cells. And when you lose receptor cells, your sense of taste doesn't remain as acuitous as when you were much younger. Just one more thing about gustation. We're going to talk about the neural pathways that carry this sensory information into our central nervous system. Neurotransmitters from receptor cells will stimulate cranial nerves to basically send impulses into our central nervous system, specifically our brain. So now you'll need to know the cranial nerves that are involved. So make sure you know these cranial nerves that are involved in sending this information to the medulla portion of our brain, where that information will be routed to the gustatory cortex and a number of other places. Those cranial nerves are 7, 9, and 10. And you, of course, at this point, know the names of those nerves. So the information is ultimately sent to the medulla and is routed to the appropriate destinations. Gustatory cortex via the thalamus. You remember that's kind of an integration place where I send impulses to the appropriate places in the brain. The hypothalamus and the limbic system, and parasympathetic activity that's going to actually innervate the digestive system in anticipation of this food that we're eating. Now, one more thing, and that is when we start talking about olfaction, which is our sense of smell that we'll take care of in the next couple slides, you will notice that the same destinations of sensory information from our nose, the sense of smell, is going to be traveling to the same basic places. Well, let's talk about olfaction, how we smell things. A lot of what we smell is very similar in terms of what we taste. As a matter of fact, the senses of taste and smell are almost indistinguishable. How's that? Well, fact of the matter is a lot of what we taste, incidentally, is perceived in our nose, not just only in our taste buds. You start chewing and the volatilization of that food material is going in and out through your nose. It's going to be crossing over these olfactory receptors. So let's talk about what we smell. What we smell is commonly referred to as odorant, and these are volatilized molecules that are going to be suspended in the air. And those volatilized molecules enter into our respiratory system and they encounter our olfactory receptors in our nasal cavity. Well, we've got this region in the upper regions of our nasal cavity where we have an olfactory epithelium, and the olfactory epithelium is covered with olfactory receptors. And those olfactory receptors connected to their individual neurons can detect somewhere near around 10,000 different odors. These molecules that we breathe, molecules that are suspended in the air, encounter our mucus, this mucus lining that's found lining the epithelium in our upper respiratory tract. And those molecules dissolved in the mucus therefore bind with these receptors, and at that point create a situation where we will stimulate a second messenger system inside of those cells. And that second messenger then opens the sodium channels and initiates a depolarization. So these aren't ion gated channels, these aren't ligand gated channels. These are situations where the receptor binds. We end up stimulating a second messenger molecule within the receptor cell. That second messenger molecule is probably something called cyclic AMP, which is similar to adenosine triphosphate. But the bottom line is that which generates a depolarization. A depolarization is going to be carried ultimately to the olfactory portion of the cerebral cortex, where we're going to interpret that as a particular smell. Now, on our next slide, we're going to talk a little bit more about the wiring, I guess we could say, of how these receptors are wired and how that information, the sensory information, is collected and routed into the brain. And it's going to be routed through a single cranial nerve. Unlike taste, which is routed through a group of 3 different cranial nerves, we're going to route all this information through one cranial nerve to the brain. Let's take a look at the wiring. If you take a look at the illustration at right, you'll see a series of many olfactory receptors. And every single one of those olfactory receptors will respond to a certain type of molecule. But all those olfactory receptors are linked together, ultimately connecting to a region that's right above the ethmoid bone. The ethmoid bone is the floor of the skull, lying between your olfactory region and this thing called the olfactory bulb, where all of these sensory neurons are going to meet and then route outwards through the olfactory nerve, cranial nerve number 1, that ultimately is going to connect to the olfactory region of the cerebral cortex. But let's see how that happens. All of these axons traveling from these receptors hook into these bulb-like structures, and you'll see them illustrated as a blue, green, red, purple, and another kind of dark greenish ball. Those are called glomeruli. And the word glomerulus literally means a ball. Later on in anatomy and physiology too, when you start talking about the function of the kidneys, you'll identify small ball-like capillary beds called glomeruli that are going to be critical to the process of filtering blood. So that being said, going back to our picture, we've got these glomeruli. They're nothing more than high concentrations of interconnections between neurons and these receptor cells. Now, the neurons that these receptor cells ultimately connect to are called mitral cells. And a mitral cell kind of sits in between the receptor and ultimately the olfactory portion of the cerebral cortex. And so all these connections are going to be made at these glomeruli. And you'll see the glomeruli synapse with these mitral cells, and that's all happening in the olfactory bulb. The olfactory bulb, like I said, sits right above the ethmoid bone, so all the connections to the receptors travel through the ethmoid bone, hook up at the olfactory bulb, and all that information is ultimately going to be routed to the olfactory cortex, that part of the cerebral cortex that's going to interpret smell. Now, you'll remember something, hopefully. We talked about taste. Taste sensation was routed through sensory neurons, and those sensory neurons carried through 3 different cranial nerves, all ultimately hooked into the thalamus. And at that point, the thalamus routed that information to the appropriate places. In this case, all of this information is going to be carried directly to the olfactory portion of the cerebral cortex. There is no thalamus involved. Once that happens, though, then the appropriate information is going to be routed to the hypothalamus and the limbic system, where we actually will remember what we smell, connect that smell with a memory or maybe with an emotion. It's going to trigger appetite and also route it to our digestive system. So I see ultimately the destinations of where this information is headed is exactly the same destination that I was heading to as far as taste is concerned. Except where taste occurred, we routed all the information through the thalamus and then sent it to these appropriate places. In this case, we're sending information directly to the olfactory cortex, and then we're sending it to the destinations of the limbic system and the digestive system. Now, there's one more thing I want to notice, and I might have mentioned this in terms of taste, but I don't think I did. So I'm going to right now. Our digestive system, when we want to control our digestive system, we can either control it through the sympathetic portion of the autonomic nervous system or through the parasympathetic portion of the autonomic nervous system. We talk about this later on, I believe in presentation number 13 or 14 maybe. We will talk about the parasympathetic versus the sympathetic portions of the autonomic nervous system. These are portions of our nervous system that will control things automatically. We don't have to think about it. But I do need you to know that the digestive system is under control of our parasympathetic portion of the autonomic nervous system. Make sure you remember that, because it's going to have importance on question or questions on future exams. Let's turn our attention to our sense of hearing. In terms of ear anatomy, I'm sure you covered a lot of this in the laboratory portion of the course. If this particular diagram, which comes from your OpenStax text, does not do it for you, please make sure you Google it so you get a much better idea of the regions of the ear and the parts and the names that you're going to need to know. You'll need to know every single bold print term on this slide. You'll need to know what constitutes the outer ear, the middle ear, and the inner ear, not only in terms of what's there, but in terms of what happens. So outer ear, pinna, which is your earlobe, the external acoustic meatus, which is the ear canal, the tympanic membrane, which is your eardrum, and inside of that is the middle ear, where we actually amplify the sound waves that we've collected in the outer ear, and we do so by sending these vibrations collected at the tympanic membrane through a series of small bones called ossicles. Those include something commonly referred to as a hammer, known as the malleus, something commonly called the anvil, technically referred to as the incus, and then finally something commonly called the stirp or the stapes. And those ultimately are connected to something called the oval window as part of the cochlea in our inner ear, where we actually take those vibrations, we're going to transduce them into action potentials that are ultimately going to be carried to the auditory cortex portion of our cerebral cortex. So make sure you know all these regions, make sure all these parts, make sure you know the names of the ossicles and their order. You'll also notice that there's something called the eustachian tube, called the pharyngotympanic tube, that actually functions as a pressure gauge between your middle ear and the outside environment. And that eustachian tube ultimately opens into the pharyngeal region of the mouth. As stated previously, that part of the ear where we actually convert vibrations into action potentials is the inner ear, where we find a structure called the cochlea. Let's take a look at the anatomy of the cochlea. The cochlea itself looks like a snail shell. Any illustration will show you the cochlea is this coiled up thing. So the best way to think about the cochlea in terms of anatomy is to uncoil it. Our slide says the cochlea is coiled. On the upper right, you'll see the illustration showing you the cochlea as a coiled up structure. And in cross-section, if I take a look at that cochlea, I notice there are three different chambers. Something called the scala vestibuli above, something called the scala tympani below, and something called the scala media, also known as the cochlear duct, smack dab in the middle. And it's the cochlear duct where we find a special organ that is going to be responsible for taking the vibrations that we initially collect at the pinny and amplify in the middle ear through those ossicles. We're going to take those vibrations and we're going to convert them into action potentials. And that structure is called the organ of Corti. Take a look at the illustration at right once again, but this time look at the bottom. You will see that we're going to unravel this cochlea. And when we unravel it, we see it does stretch out into a linear structure. And looking at the linear structure in cross-section, I see the scala vestibuli above, the scala tympani below, and the scala media in between. Now, it doesn't show it real well, but I do need you to know at the very distal end of the cochlea, you run all the way to the very end of the cochlea, the scala vestibuli, which is the cavity above, and the scala tympani, which is the cavity below, interconnect. The interconnection at the very end of the cochlea is called the helicotrema. That is spelled Helicotrema. Helicotrema. It's not listed on our slide, and it's not labeled, and it's not illustrated on this slide, and I don't think it's labeled or illustrated anywhere else. But it's extremely important because these vibrations that we are going to be collecting at the oval window, the same vibrations that we collect from the pinna, the same vibrations that we amplify through the ossicles in the middle ear, is going to be vibrating through the scala vestibuli, and the scala vestibuli is full of a fluid, and that fluid is called perilymph. And the scala tympani below is also filled with perilymph. They're interconnected, and the sound waves are going to start moving through this fluid. And so the sound waves will move down through the scala vestibuli and then will round through the helicotrema and then come back through the scala tympani. And ultimately, those waves passing through that fluid will cause some vibration to occur in the middle portion of the cochlea, what we call the scala media. And when that thing starts vibrating, that's how we're actually going to generate some action potentials. And those action potentials are going to be collected and transmitted to the auditory cortex portion of the cerebral cortex, where we're going to be able to interpret that. But more about that in a little bit. Now I want to take a closer look at this cochlea and a closer look in terms of this thing that I called the organ of Corti. The organ of Corti stretches the entire length of the scala media. The scala media, remember, that is the cavity found in the middle. Now, the organ of Corti is consisting of cells that are found on something called the basilar membrane. The basilar membrane is that part of the cochlea that separates the scala tympani from the scala media. So in other words, the scala media, which is in the middle, is subtended by something called the basilar membrane, which is found immediately above the scala tympani. And this basilar membrane contains a whole bunch of cells called hair cells. Now, these hair cells have tiny little hairs that are officially known as stereocilia. And the stereocilia project upwards from the basilar membrane, and they connect to another layer of tissue called the tectorial membrane. And the tectorial membrane lies immediately above the basilar membrane and also runs along the entire length of the scala media. So the fact of the matter is, when these sound waves travel through the perilymph of the scala vestibuli and travel through the perilymph of the scala tympani, the tectorial membrane is going to vibrate, the basilar membrane is going to vibrate, the hair cells are going to vibrate. And that's going to cause deformations in those teeny tiny little stereocilia, because the stereocilia are connected ultimately to the tectorial membrane. So there's a difference in vibration going on between the basilar membrane and the tectorial membrane above. And when you get a deformation of the stereocilia, you will get depolarizations occurring, and those depolarizations will generate action potentials that will ultimately be collected along the length of these neurons and sent to the auditory cortex via the auditory nerve. All right, I know that was a lot on our previous slide, but what I'm going to do is I'm going to take this opportunity for this slide to connect the anatomy with the actual process. And I've got some nice illustrations here to show me the transmission of sound waves to and through the cochlea. So you'll see our illustration of the ear at top. I see the pinna, I see the external auditory meatus, I see the tympanic membrane, I see the middle ear where the vibrations are amplified across the ossicles, the malleus, the incus, and the stapes, and that's ultimately connected to the oval window of the cochlea. Follow the arrow and I see a close-up view of the cochlea. I see these vibrations which have been collected by the pinna, which have been amplified by the ossicles, are now traveling down the length of the scala vestibuli, and can't see it, but they're going to go all the way to the end of the cochlea. They're going to make a U-turn where I find the helicotrema, not illustrated there, but they're going to come back. And if I take a look at the scala tympani below, I see the returning sound waves. And in between the two, I see, illustrated in blue, in the middle is my cochlear duct, i.e. the scala media. And in the cochlear duct, I see my organ of Corti stretching the entire length of this unraveled cochlea. And that's where pressure from the sound waves traveling down the scala vestibuli are going to be causing vibrations in the tectorial membrane, and the vibrations in the tectorial membrane offset by vibrations in the basilar membrane are going to cause deformations of those hair cells, and those hair cells are going to generate the depolarizations, going to generate the action potentials that are going to be carried to our brain via our auditory nerve. That's how we convert the transmission of sound waves to and through the cochlea into action potentials that are going to ultimately give us an ability to determine what we hear. Can I say one more time? Our slide, auditory pathway here, is going to rehearse some of this information we've already covered. It's also going to give us some additional information, and the additional information has to do with how we actually generate the action potentials and how we distinguish between loud sounds, small sounds, high pitches, and low pitches. So let me run down the bullet points here. What's very convenient is I've got this cross-sectional view of my cochlea at right. I see the scala vestibuli, I see the scala tympani, I see the scala media in between, I see the close-up view of the organ of Corti, and down below I see the basilar membrane, I see hair cells in yellow, I see stereocilia attached to the tectorial membrane above. Let's look at the bullet points. The oval window vibrates. The fluid in the cochlear duct vibrates. The basilar membrane has hair cells. We call those stereocilia. The stereocilia move with the vibration. Remember, it's a vibration occurring in the tectorial membrane that's offset by the vibration in the basilar membrane, and this causes ion channels to open in those hair cells. That depolarizes the cells. The information is sent down the auditory nerve. Loud sound, more hair cells being stimulated. Small sound, fewer hair cells being stimulated. High-pitched noises are generally going to move hair cells present at the base or the proximal section of the cochlea, whereas low pitch will wind up moving cells more distally of the cochlea. So the distance traveled through the cochlea is going to be much greater for low-pitched vibrations versus high-pitched vibrations. And that's how we interpret pitch. And now for the details in conducting all this auditory information into the brain. We have the collected depolarizations and action potentials generated from hair cells in the organ of Corti, and those are collected within something called cranial nerve 8, also known as the vestibulocochlear nerve, also known as the auditory nerve, and sometimes known as the acoustic nerve. Regardless of its name, it's strictly a sensory cranial nerve, and it arrives at the medulla. And at that point, it specifically gives its branches off to something called the superior olivary complex, which is just nothing more than some nuclei located on either side of the pons. And I see them in our illustration there. They're kind of illustrated as nuclei in red. And those are going to be carried into the midbrain. Now, the thing is looking at this illustration, you will notice that there seems to be crossover from the left side to the right side. Now, we had said once upon a time that was contralateral connections. And so our cochlear tracts, or our nerves, coming from the left ear and from the right ear, crisscross. And the right side goes to the left brain, and the left side goes to the right brain. But in some cases, some of those tracts are on the same side, hence we use the word ipsilateral. So bottom line is, as these sensory auditory tracts come into the brain, they connect with the superior olivary complex, and some of those connect with the same side and some of those connect with the opposite side. Regardless, once they come in, they connect to neurons that are going to be found in the inferior colliculi. Once again, another set of nuclei found, in this case, in the midbrain. And here's where we get connections with our motor neurons, portion of the somatic nervous system, and limbic connections that go to the place where we deal with emotions and memories. Now, in the midbrain, we also have tracts that are going to carry this same information into the thalamus. And a part of the thalamus that's critical here is something called the medial geniculate body. The medial geniculate body technically is part of the thalamus, and it is responsible for processing sound localization and speech interpretation. In other words, it's going to basically do all the routing that's going to be necessary for us to interpret this sound that's coming into our brain. Finally, we arrive at the primary auditory cortex, which is located in the temporal lobe. Ultimately, we're able to identify what the sound is, we're able to interpret it, we're able to identify where it came from, we're able to access memory and emotions in a means of being able to react to it in an appropriate manner. So, in summary, sensory information coming from the hair cells in the organ of Corti, located in the right ear, in the right cochlea, and in the left ear, in the left cochlea, are going to be sending those sensory impulses into the brain via the vestibulocochlear nerve, also known as the auditory nerve, also known as the acoustic nerve, also known as cranial nerve number 8. It arrives at the brainstem. And when it does, it enters more or less bilaterally where we find the pons. And it connects with these two regions of brain nuclei called the superior olivary complex. And at that point, those tracts will be referred to the inferior colliculi in the midbrain, at which place we will actually connect to motor regions and limbic regions, and ultimately connect to the thalamus, where we will process the sound, where it's coming from, and being able to interpret speech, and then finally, ultimately, to the primary auditory cortex. Put all together, we get it all figured out. Now, also, we mentioned the fact, and it is important, that some of this information from, for example, the right ear will travel to the left side of the brain, whereas some of the information will stay on the right side of the brain. Some information from the left ear will go to the right side of the brain, and some will stay on the left. Hence the difference between what constitutes contralateral versus ipsilateral connections. All right, let's talk about the vestibular system, which monitors equilibrium. And it's also located right alongside of the cochlea. So let's see what it assesses. It assesses static position, which is your non-moving position. I'm sitting in front of a microphone right at this minute. I'm vertically oriented, and I know I'm vertically oriented, and I know that I'm seated. And all of that is because of information being fed into my brain from my vestibular system. One unit of that system found on the right-hand side of my head and the other found on the left-hand side of my head. Now, if I stand up real quick, I'm aware of that too. That's linear acceleration. That's acceleration in one vector direction, upwards. If I get on an elevator, it sends me up. I'm aware of that fact. If the elevator goes down, I'm going down, and I'm aware of that fact. That's vertical acceleration. On the other hand, if I get in my car and I drive real fast in one direction, I'm aware of that. And if I put on the brakes and I stop suddenly, I'm aware of that. That's horizontal acceleration. That's all taken care of in a portion of my vestibular system called the vestibule. There are two parts to it, one called the utricle and one called the saccule, but we'll get into that in a little bit. Now you'll notice over there on the right-hand side of the slide an illustration of the ear and a close-up view of your vestibular system. You got one of these on each side of your head. And yes, the vestibular system appears to be attached to the cochlea. And I notice that the vestibular system consists of the vestibule, consisting of the utricle and the saccule that I just mentioned. And that's what's dealing with your monitoring of static position and linear acceleration, whether that be vertical or be horizontal. But then I've got semicircular canals. And the semicircular canals are going to monitor positions of your head. And that's extremely important because if you think about it, if something's coming at you from the right and you turn, or something's coming you from the left and you turn, or a plane is going overhead and you turn and you see that, or you feel something bump against your leg and it's your cat, but you look down, and in each of these cases you're making rotary movements of your head. Now, you can probably imagine the complexity of all of the processing decisions that need to be made because you change the positions of your head every day on a regular basis, and you do it very, very quickly. And every time you change the position of your head, you're well aware of the fact of where your head is with respect to your body. You don't fall over because you lose your balance. Everything is fine. So the vestibular system consists of two units, one on the left side, one on the right side. They are associated with your inner ear. They're connected to the cochlea. They consist of something called the vestibule. They consist of something called the semicircular canals. And you'll need to know what those two portions of the vestibular unit are going to be involved in monitoring. Now let's talk about the details. We are going to start off by looking at the receptor structures that are located in the vestibule. These receptor structures are called maculae. There's one macula found in the utricle and one macula found in the saccule. Talk about those in a second. But looking up close at a macula, it's nothing more than a small field or arrangement of hair cells. These hair cells look an awful lot like the hair cells that we saw in the organ of Corti. As a matter of fact, they've got little hairs that are called stereocilia, and they're going to operate the same way. If you deform a hair, you can create a situation, in this case, where you open mechanically gated channels, but initiate a depolarization. And so that depolarization is going to result in an action potential that represents some amount of information that's going to be carried to the vestibular cortex of your brain where you can interpret it. So let's take a look at the picture. I've got these hair cells, and they're actually embedded in a layer of gelatinous material, and that gelatinous material is topped with an arrangement of small calcium carbonate crystals called otoliths. Sometimes they're collectively referred to as stytoconia. But the fact of the matter is, if you suddenly change your position, you're moving forward and you stop. You're moving backward and you stop. You bend over to pick something up. You turn yourself upright because you're finished bending over. In each of those cases, that gelatinous material is going to shift. And it's going to shift because the otoliths have mass and they shift and the gelatinous material shifts, which means you've got a mechanical deformation taking place in the gelatinous material, and that deformation is going to be imparted on those stereocilia. And as soon as you create that deformation in the stereocilia, it's going to create a depolarization because you're going to open mechanically gated channels. All of a sudden, the sodium comes rushing in, the potassium goes rushing out, and you've got a depolarization. And that depolarization very quickly can become an action potential, and off it goes. We're about to contrast the utricle and the saccule as parts of the vestibule. What I want to first do is take a look at the pen and ink drawing at the lower right, and you'll see the anatomy of our inner ear. You'll see the cochlea, but then you see the vestibular apparatus. The vestibular apparatus consists of the vestibule, and the vestibule has two parts. One called the saccule, which is proximal to the cochlea, and the second is the utricle, which is distal. Or, if looking at this picture, I notice that the saccule is below the utricle. And the utricle is actually located right next to the origins of the semicircular canals. And I'll see there's three of those. One called the horizontal canal, one called the posterior canal, and one called the superior canal. But at this point, we are going to be focusing on the utricle and the saccule in the vestibule. Now you'll remember the vestibule monitors static position, monitors horizontal acceleration, and monitors vertical acceleration. The utricle, which is located right next to the semicircular canals, is located distally from the cochlea, has hair cells in a macula that happen to be vertically oriented. So you'll see in the color illustration that the hair cells are in fact oriented vertically. They are embedded in that gelatinous layer, and I see the otoliths lying above. And I notice that the individual in this illustration is bending forward, and that's deforming the gelatinous matrix because the otoliths are shifting forward, and that's going to deform the hair cells. Now, the utricle itself contains a fluid called endolymph. So it's actually the movement of the body shifts the endolymph, which in turn shifts these otoliths. But regardless, it's the mass of the otoliths, as said before, that actually causes the deformation in the gelatinous layer, that causes a deformation of the hair cells, generates the depolarizations, and generates the action potentials. Now that we're done with the utricle, let's take a look at the saccule. The saccule's job is to monitor vertical linear acceleration. As a consequence, you would expect that probably these hair cells would have a different orientation. And I see, yes, they are horizontally oriented. In the utricle, they were vertically oriented. Now they're horizontally oriented. The whole point, if we're going to assess vertical acceleration, I get up, I sit down, I go up an elevator, I go down an elevator. That's going to be monitored by shifts in those otoliths. And once again, that's going to be the shifting of the otoliths in a fluid called endolymph. The endolymph shifts, the otolith shifts, the gelatinous matrix shifts, and I get a deformation of the hair cells. And once I deform those hair cells, that's going to generate depolarizations, that's going to create action potentials, and all of this information is going to travel through cranial nerve 8, the vestibulocochlear nerve to the vestibular cortex of the brain, where it's all going to be interpreted. But once again, that's not all. We got to talk about these semicircular canals. Last but not least, in this presentation on senses, we're going to talk about vision, and we're going to begin with the anatomy of the eye. And I am sure, once again, you probably covered this in the lab portion of the course. This particular illustration is just a run-of-the-mill anatomical assessment of the structure of the eye. If you don't groove on this, go on Google, get another one. Every single structure on this slide is fair game. Now, I will be honest with you, I have no idea why some of those structures are highlighted with a yellow shaded box. It's one of the situations where I inherited this presentation and I'm not quite sure what that is. So make sure you know all of the structures here. We will cover a significant number of them, and if they have a function, we'll be sure to identify that on the next series of slides. Just right now, make sure you are aware of these structures. And if you have any question at all as to what they do or what they're made of, please Google each one. That might not be a bad idea anyway, and I'm sure they're all documented in that OpenStax book of yours. But like for me, I find it much easier to get my information from a Google search rather than actually try to read a darn book, because I'm so ADD, I can't even focus on the darn thing because I wind up reading the same sentence 3 times over. I am a big fan of Google, and I'm a big fan of Wikipedia. I have no problem with you doing that because essentially the more knowledge you get, the better off you're going to be. So I am not going to dwell on this slide much beyond to say to you, if I were you, I would make sure I know every single one of these structures. And they are going to have, at least most of them, are going to have some import on some future slide in just a little bit. Receptor structures in the semicircular canals are found in the ampullae found at the base. Each semicircular canal features one ampulla. And we're going to see in a little bit that the structure, that is the receptor structure, is really, really similar to those maculae that we found in the utricle and the saccule. The sensory structure in each semicircular canal is called a cupula. And the cupula, if you take a look at the illustration there, is nothing more than this little blob of gelatinous material. And that blob of gelatinous material is going to sit on top of a bunch of hair cells. So the semicircular canal is filled with endolymph. You turn your head. The endolymph shifts. The rotational movement moves the endolymph, which moves the cupula, which in turn moves and deforms the hair cells. In turn, generates a depolarization. In turn, generates an action potential, which travels through the vestibular cochlear nerve to the vestibular cortex, where it's going to be interpreted. Last but not least, in this presentation on senses, we're going to talk about vision, and we're going to begin with the anatomy of the eye. And I am sure, once again, you probably covered this in the lab portion of the course. This particular illustration is just a run-of-the-mill anatomical assessment of the structure of the eye. If you don't groove on this, go on Google, get another one. Every single structure on this slide is fair game. Now, I will be honest with you, I have no idea why some of those structures are highlighted with a yellow shaded box. It's one of the situations where I inherited this presentation and I'm not quite sure what that is. So make sure you know all of the structures here. We will cover a significant number of them, and if they have a function, we'll be sure to identify that on the next series of slides. Just right now, make sure you are aware of these structures. And if you have any question at all as to what they do or what they're made of, please Google each one. That might not be a bad idea anyway. And I'm sure they're all documented in that OpenStax book of yours. But like for me, I find it much easier to get my information from a Google search rather than actually try to read a darn book, because I'm so ADD I can't even focus on the darn thing because I wind up reading the same sentence 3 times over. I am a big fan of Google, and I'm a big fan of Wikipedia. I have no problem with you doing that because essentially the more knowledge you get, the better off you're going to be. So I am not going to dwell on this slide much beyond to say to you, if I were you, I would make sure I know every single one of these structures. And they are going to have, at least most of them, are going to have some import on some future slide in just a little bit. Okay, so thinking of that illustration on the previous slide, we're going to observe the fact that the eyeball is actually a laminate structure, meaning it consists of a number of different layers. Now, those layers from the outside to the inside consist of the fibrous layer, the outermost layer, the vascular layer, smack dab in the middle, the neural layer, which is found on the inside. Now, in each of these cases, I see that the outer layer of the eye, the fibrous layer, sometimes called the fibrous tunic, is essentially consisting of the sclera, which is the white portion of the eye, and the cornea, which is that clear portion covering the front. And I notice that I've got pain receptors in the sclera, and I notice that the cornea consists of stratified squamous epithelium, at least on the outer layer. It is innervated, so it will hurt if you get it scratched, and it is responsible for a visual process known as refraction, and we'll talk more about that in a little bit. Just inside of the fibrous layer is our vascular layer, which consists of the choroid, which consists of a dense amount of blood vessels that provide oxygen and nutrients to the cells of the eye, a ciliary body, which consists of a number of ciliary muscles and suspensory ligaments that hold the lens in place, and then the iris, the colored portion of the eye, including the opening or the aperture called the pupil. And so in that case, we're regulating the amount of light that enters the eye, and obviously the iris is the pigmented portion of the eye, which gives you your eye color. And then finally, the innermost layer is the retina, which is primarily concentrated on the back wall of the eyeball, where we find our photoreceptors, both rods and cones. We'll talk more about those in a little bit. And that's where we find a connection to the optic nerve, which is going to carry this visual information from our eye to our visual cortex. Well, the eyeball is a hollow structure too, and it's got 2 cavities, an anterior cavity and a posterior cavity. Well, the anterior cavity is found in front of the lens, and the most conspicuous structure within the anterior cavity is going to be the iris. Now, just a comment about the iris. I mentioned before that its job is to regulate the diameter of the pupil. The pupil is an aperture. It's an opening. It's an opening through which light enters the eye, and that light is ultimately going to be projected on the back wall of the eyeball, where all of our photoreceptors are going to be. And so our iris's job is going to be to regulate the size of that pupil. There is a muscle associated with that. That muscle is called the iris sphincter, or sometimes it's also referred to as the pupillary sphincter. And that sphincter muscle is a circular muscle, as all sphincter muscles are, and it's located right about the periphery of the pupil. When that sphincter muscle contracts, the pupil constricts. And when the sphincter muscle relaxes, the pupil dilates. The anterior chamber at the very front is the cornea. So the anterior chamber occurs between the cornea and the lens, and it's full of a clear fluid called the aqueous humor. Now, if you take a look at our slide, the subpoints under anterior cavity with aqueous humor, blood supply to lens, anterior and posterior chambers, glaucoma, all those statements have nothing to do with the anterior cavity. I don't know why they're there. It looks like somebody made the slide and got distracted, but make sure you omit blood supply to lens, anterior and posterior chambers, and glaucoma. Glaucoma has nothing directly to do with the anterior cavity. Neither does blood supply to the lens, and neither does anterior and posterior chambers. Now, the other portion of the eye, which is a much larger chamber in the eye, is called the posterior cavity. And the posterior cavity is bounded on the front by the lens, and it's bounded on the back end by the retina. So the far majority of the volume of the eye is occupied by what we call the posterior cavity, and it's full of a viscous, thick fluid that's more or less brownish in color called the vitreous humor. And even though it's brownish in color, it is going to permit the transmission of light. Because understand, the job of the eye is to collect light from outside the body, focus it, and project it on these photoreceptors that are going to be located on the retina. And once again, support, yeah, vaguely, yes, the vitreous humor is going to provide physical support for the eyeball maintaining its spherical shape. Embryonic development, I'm not quite sure what that refers to. If you want to find out more about the anterior cavity or the posterior cavity, please refer to your book or please refer to a Google search. But at this point, all I need you to know is the anterior cavity is found between the lens and the cornea. It's characterized by the iris. It's characterized by aqueous humor. The posterior cavity occurs posterior to the lens, between the lens and retina, and it's full of a viscous brownish colored fluid called the vitreous humor. And sometimes, I will tell you this, sometimes the vitreous humor is also known as the vitreous body. While I am not going to specifically cover the accessory eye structures mentioned on this slide, you'll need to know them. In particular, make sure you know the role of the conjunctiva, and in particular, know what conjunctivitis happens to be. Also commonly known as pink eye is an inflammation of this pretty darn important portion of the eye structure. Let's talk about tears. Tears are generated by something called the lacrimal gland. There's one located above and lateral to the eyeball on each side. And the lacrimal gland is an exocrine gland. And so it's going to be drained by a lacrimal duct. The lacrimal duct is going to release those tears at the top of the eye, and they're going to flow across the eye surface. And this is amazing to me. I never realized this, is that when those tears flow across the surface of the eye, they are ultimately going to be collected by something called the lacrimal canaliculi, which is nothing more than a teeny tiny little canal that is going to be draining into the nose via something called the nasolacrimal duct. So you got this method of constantly getting tears across the eye, and those tears are constantly being drained out, so therefore you're not going to be tearing constantly. The tears flow in, the tears flow out, ultimately into the nose via something called a nasolacrimal duct. The composition of tears are primarily water, but there are going to contain some lipids, something called lysozyme, which is a body defense type of substance that can disrupt bacterial cell walls, something called lipocalin, which actually is a protein that captures hydrophobic molecules that shouldn't be there, and it can basically take care of those. There's going to be some glucose, there's going to be some sodium as well. So the lacrimal gland generates tears. Tears are primarily going to be involved in lubrication of the eyeball, so you don't scratch your cornea through constant blinking of the eyelids. The fact of the matter is, it's also going to have a somewhat defensive role as well. Let's take a look at the organization of the neural layer, sometimes called the neural tunic. Now, you'll remember that the posterior wall of the eye, that would be the posterior wall of the posterior chamber, is the retina, and it's lined with photoreceptors. But what's unique about the setup is the photoreceptors, which are actually collecting the light and generating depolarizations because of that light, are ultimately going to be connected to a bunch of neurons that will interconnect with other neurons that will interconnect with other neurons that will eventually create the optic nerve that will carry all of this photoreceptor information to the visual cortex of the brain. But interestingly enough, if you take a look at our illustration, you'll notice that the light is shining on the retina, but what's on the surface of the retina are all of the neurons, all the axons. It's the photoreceptors that are actually located behind those. So as the light is striking the retina, it's not striking the photoreceptors immediately, it's striking the cells, the neurons that are ultimately connected to those receptor cells. So you would take a look at the small illustration at the top right, and you'll see the little yellow arrow indicating the direction of light passing through a series of cells and more cells and finally getting to those photoreceptors. So hence the bullet point that says backwards setup. The photoreceptors are found on the back side of the retina, not on the front. And the photoreceptors are going to consist of two different types, rods and cones. We'll talk more about them in a little bit. Now, there is a place on the retina called the macula lutea. And the macula lutea is what makes our vision so acute as far as our ability to discern color and our ability to discern detail. And it's our vision found smack dab in the center. So right now I'm looking at my computer screen, and if I'm looking straight ahead, I see very accurately the illustration of the eye, the illustration of the retina, and all the little labels. But if I essentially try to see to the right of that, above that, to the left, below that, the visual acuity becomes a little bit less. So what happens is our eye is going to be involved in focusing the far majority of what we look at on that one place on our retina called the macula lutea, sometimes also called the fovea. And the fovea is where we have this ultra high concentration of a type of photoreceptor called a cone. And that's what gives us our detailed central vision. So our photoreceptors, those rods and those cones, connect to bipolar cells, which is nothing more than a neuron that has two poles. One pole connected to the receptor and the other pole connected to a ganglion cell. And these ganglion cells collectively form the optic nerve. So all of this disparate photoreceptor information coming in through all these cones and all these rods connect to bipolar cells that in turn connect to ganglia cells that in turn connect to the optic nerve. And the optic nerve, its physical location represents a blind spot on the retina because obviously there are no photoreceptors where that occurs. Now, a lot of times what happens if there's missing visual information coming in from that eye, the other eye, because we're looking at things stereoscopically, is going to compensate for that. So our brain has the ability to compensate for the fact that there is a hole in the vision of our right eye and the left eye simply by virtue of the fact that the optic nerve takes up some of that space and there aren't any photoreceptors there. We've kind of studiously avoided the lens. We understand it's like the dividing line between the anterior and posterior chamber. And most of us know that the lens is going to be involved in focusing light somehow, but how is it involved? I mean, what is it made of? Well, let's take a look at the lens. First off, it tells me, yes, the lens will focus light. And the process of focusing light is called accommodation. So what's going to happen is when you're looking at something really up close, the lens can change its shape. And in changing its shape, it can focus light on the retina in a specific place, in a specific clarity. It's like when you adjust the focus on a projector. You're adjusting the focus so the image is not blurry. You want to make it so that the focal point of the image is right there on the screen. Adjusting the lens on a projector is accommodation. That's what your lens does. If you take a look at pictures here, you'll see that the lens does change shape. You'll remember there's this thing called the ciliary body, and there's these things called ciliary muscles, and those ciliary muscles are attached to the lens around its periphery, and when they contract, they will pull on the lens. And you can pull on the lens and make it not quite as fat as it was previously, and sometimes the lens can get big and fat. So the lens itself is avascular. There are no blood vessels. It does get oxygen and nutrients from the surrounding fluid. In this case, it's going to be aqueous humor primarily, and it does maintain itself through regular mitotic activity. Now, most of us have heard of cataracts. Well, the lens is primarily protein in its composition, but the fact of the matter is if those proteins start breaking down, then the lens becomes clouded, and that's what the process of forming a cataract is involved with. We're about to talk about focus issues, but what's nice about this slide is it's going to contrast what the lens does with what the cornea does. Now, I talked about the cornea, what seems like many, many slides ago. Said the cornea was that clear portion of the fibrous tunic that exists at the front side of the anterior chamber. The cornea has a contribution to make to keeping your vision in focus. You'll remember that we can actually do something called accommodation with the lens. The ciliary muscles can exert contractive force on the lens, changing its shape. I change the shape of the lens, I can change the focal point of the image and hopefully project that image in a very, very clear way exactly on the retina where I need to. But the cornea participates in that as well in a process known as refraction. And refraction is nothing more than bending the light rays so they can actually approach the lens at an appropriate angle. Accommodation is adjustable. Refraction really is not. But the combination of refraction and accommodation allows us to make sure that that image is projected on our retina where it's supposed to be. Refraction is our initial adjustment of those light rays. Accommodation is how we manage those as we move closer or further away from the object that we are looking at. But now what we're going to talk about is focus. And there are some focus issues that we associate with the eye. I'm nearsighted. I've got a brother who's farsighted. My wife has got an astigmatism. So does one of my kids. What are these visual disturbances? Well, we're going to find in a minute that they have something to do with the shape of our eye and how that actually influences our ability to create a focal point. Now, ideally, perfect vision would place that focal point right at the retina, not in front of it, not behind. Because as long as that focal point is right on the retina, the image is going to be in perfect focus. It's going to be clear, it's going to be sharp. So let's take a look at these illustrations on the right-hand side of our slide. I'm looking at a candle. Now you will notice in the normal vision illustration, as I'm looking at the candle's vertical. A consequence of the way our vision works is the candle's image gets inverted, gets turned upside down. And so what's actually presented to our retina is an upside down image. Our brain is responsible for interpreting that image as being right side up. Thank goodness for our brain, because right now you're looking at everything upside down. At least that's the way it's being projected on your retina. But take a look at that top illustration. You'll see that the image of the candle, even though it's upside down, is being focused on the fovea of the retina, smack dab in the middle of the retina, and you'll notice the image is being projected right at the point of where the retina is. But let's take a look at myopia. In myopia, your eyeball is relatively long. And because of that, what happens is the image of the candle, once again, is being projected invertedly, but it's actually, the focal point is in front of the retina. So you're projecting the focused image of the candle some distance in front of the retina, which means the actual image that's landing on the retina is not going to be quite as clear, and it's going to be a little bit fuzzy, and it's going to be out of focus. Myopia is sometimes also referred to as nearsightedness, and it's generally a consequence of having an eyeball that's a little bit too long. So what we need to do is we're going to need to take that focal point and move it further into the distance. In order to do that, I need a concave lens. If you look at the very last illustration, a concave lens is actually going to spread the light rays out a little bit and actually allow us to move the focal point of the candle further off in distance. So I'm nearsighted, and if I take a look at my lenses on my glasses, they are shaped very much like the lens I see in the last illustration. So, normal vision, perfect focus on the retina. Myopia or nearsightedness, focusing the image in front of the retina. Correction with a concave lens that spreads the light rays and moves the focal point back a little bit. So now the focal point is exactly on the retina itself. Now, if you take a look at the bold print term hyperopia there on our slide, that's farsightedness. That's when you have a short eyeball. That's when you're actually projecting the image behind the retina. And you can't possibly focus it behind the retina, but the fact of the matter is if that focal point is behind the retina, the image that actually is showing up on the retina is going to be every bit as fuzzy as the one in the long eyeball situation. But in this case, we're going to use a convex lens to correct that image. So there's a mistake on our slide. Under hyperopia, short eyeball, it says concave lens correction. Please change that. It should now read convex lens correction. Because what we're going to need to do is we're going to need to try to move that focal point closer to the front of the eye. Now finally, the last bullet point on our slide concerns something called astigmatism. And astigmatism occurs when your lens is of an irregular shape. So sometimes you might perceive an image as being in focus, but sometimes it might not be in focus depending on what angle the light rays are entering the lens. And so special adaptations in a lens can actually create a situation where we see much better than we would otherwise. But astigmatism is a little bit more complicated an issue than simple nearsightedness or farsightedness. So this is nice. We have some illustrations for hyperopia on the left, and there I see the normal vision illustration. There I see the hyperopia showing me that the focal point of the image is behind the retina. I see correction with a convex lens, concentrating the light rays so that I actually move the focal point closer, and therefore I actually am projecting the in-focus image right on the retina where it needs to be. And then I see a series of illustrations showing me the whole deal concerning astigmatism. Vision in an eyeball with astigmatism shows me that because of the irregular shape of the lens, that image is going to be a little off. And notice that it's the left-hand side of the image is a lot more blurry than the right, considering the fact that as that light rays passes through an unevenly shaped lens, the portion that's traveling through the uneven shaped region is going to be out of focus, whereas the portion that's traveling through the even shape is going to be a little bit better. Make sure you know what is myopia or nearsightedness, how it's corrected. Make sure you know what is hyperopia or farsightedness and how it's corrected. And definitely know what is astigmatism. Okay, ladies and gentlemen, we arrive at a slide and I have no idea why it's here. It probably should have shown up earlier in this particular presentation, or perhaps it should have shown up when we were talking about the central nervous system. Regardless, I need to cover this information. And I apologize, it's out of order, but once again, I have inherited this presentation. I do the best I can. We're going to take a look at what are called ascending sensory pathways. These are sensory pathways that are going to be collected from our various receptors in our peripheral nervous system. And those receptors are going to be carrying information into our spinal cord. And that information is going to enter into some white matter tracts that are going to be running through our spinal cord upward, upward, upward to our brain. So in each of these cases, we're going to be talking about afferent neurons. Remember, afferent neurons carry sensory information into the central nervous system. There's two ways this happens. One's called the dorsal column system, which is going to involve ipsilateral transmission of sensory information. In other words, if it comes in on the right side of the body, it's going to stay on the right-hand side of the spinal cord. It's going to go all the way up, and it might wind up into the right side of the brain. So this dorsal column system is going to be featured on the left side of our slide. On the right-hand side of our slide, we've got something called the spinothalamic tract. It is also an ascending tract. It's also going to deal with afferent neurons. It's going to be a little bit different because it's going to be characterized by contralateral transmission of sensory information. In other words, it's going to come in on the left and it's going to wind up heading to the right. It's going to go to the right and head to the left-hand side of the brain. So we're going to take a look at each of these. We're going to understand they have their own unique characteristics. They will all involve three different neurons, and they will characterize transmission of certain types of information in the body. You will need to know which of the two is contralateral, which of the two is ipsilateral, and which of the two deals with the specific different sensations we will be discussing. So let's take a look at dorsal column system, the ipsilateral transmission of sensory information. I'm looking at the illustration on the left, and I see there are afferent neurons that are going to be funneling information into two bundles, the fasciculus gracilis and the fasciculus cuneatum. I'm not worried that you can distinguish between the two. It doesn't matter. The bottom line is all of this sensory information comes into those. These are white matter tracts that are going to run the entire length of the spinal cord, ultimately to the thalamus of the brain, which you'll remember is the primary place where we route all of this sensory information to the appropriate portion of the cerebral cortex. But let's take a look at the neurons that are involved. I notice that there are going to be a first order neuron that carries the information into the spinal cord, where we will actually synapse with a second order neuron that will actually carry the information to the thalamus. At that point, a third order neuron, that is number 3 in the list, will carry the information to the appropriate cortex. So there are going to be 3 afferent neurons involved. One afferent neuron carrying the information directly into the spinal cord, another carrying the information to the thalamus, and the third carrying the information to the appropriate portion of the cerebral cortex. Now, I noticed that what's involved here would be our ability to discern fine touch. 2-point discrimination is if I take 2 sharp pins and I poke you, and those 2 sharp pins are close distance to one another, can you distinguish between 1 point or 2? Obviously, the further I move them apart, that winds up being 2-point discrimination. At some point, you will distinguish them as being two different sensations. Conscious proprioception, in other words, your balance and your position of your body and your detection of vibration, are all managed by this dorsal column system. And once again, it's all going to be ipsilateral transmission of information. What comes in on the right travels on the right. What comes in on the left travels on the left. So let's take a look at spinothalamic transmission. This is going to be contralateral. So once again, we got afferent neurons entering the spinal cord. They're going to synapse with a second order neuron. That second order neuron is going to enter a lateral spinothalamic tract. Once again, it's white matter found on, in this case, the opposite side. So it's contralateral. If it comes in on the right, it travels up the left. If it comes in on the left, it travels up the right. That second order neuron ultimately connects directly to the thalamus, where, as before, a third order neuron is going to carry that information to the appropriate cortex. But this time, it's on the opposite side of the body. So I see pain, temperature, and coarse touch. If you're experiencing pain on the right side, it's going to travel to the left. If you experience pain on the left side, it's going to be processed on the right. So make sure you know the distinction between these two different pathways of spinal transmission. One being ipsilateral, one being contralateral, one being the dorsal column system, and the other being the spinothalamic tract. And each of them governs specific transmission of specific information, and you'll need to know that. Well, this is the actual slide that we should be finishing off with as far as sight is concerned. I'm going to talk about the visual pathway. That's how we get the sensory information into the brain. And it's somewhat similar to how we dealt with auditory information. So let's take a quick look. The illustration is a little bit different from what we've seen before, but I see a couple of eyes there, and I see pathways bringing information from the eyes into the brain. We already know that we've got these cranial nerves called optic nerves, and they qualify as cranial nerve Roman numeral 2. And that information travels into the brain, and it encounters a crisscross, and that crisscross is called the optic chiasm. The optic chiasm is going to carry the image from the left eye to the right side of the brain, from the right eye to the left side of the brain, and there's going to be some crosstalk between the two of them. So there's some of that connection is going to be ipsilateral, some's going to be contralateral, but the bottom line is that information is going to be carried into the thalamic region of the brain. Now, when it gets to the thalamic region of the brain, it's going to be entertained by some nuclei found in that region of the brain. You might remember something called the inferior colliculus as far as the auditory system is concerned. In vision, that information enters something called the superior colliculus. You might remember something called the medial geniculate body in the thalamus that was processing auditory information. Well, in the visual system, it's the lateral geniculate body. So, in other words, auditory information is going to be processed by one set of colliculi and one geniculate body, whereas the visual system is going to be also managed by a colliculus and its own geniculate body as well. But the bottom line is this information is going to be processed in very much the same way. You remember the auditory pathway routed that information to the auditory cortex, but also involved connections to the limbic system and also connections to a variety of other places in the brain, because our response to an auditory sensation is much more than just the sound. It's interpreting the sound, and it's locating the sound. Well, the same thing's true with vision. See, what we're doing is we're encountering the sensory information that is going to be put together as a picture, so to speak, but it also has to be connected with what we know about what we're looking at and how we're going to respond to it. So, in much the same way, optic sensation is processed somewhat the same way as auditory sensation is. It's just a matter of routing it to the appropriate cerebral cortex. Auditory pathway routes it to the auditory cortex, and the optic pathway carries it to the visual cortex.